Thanks to everybody for coming for this presentation. So we're going to talk about a different way for testing adverse reactions to foods, different from skin testing, different from serological testing like IgE or IgG. We're going to talk about a cellular test that's been developed at Cell Science Systems and some other cellular assays that are related to identification of micronutrient deficiencies. And we'll also leave some time for some of the other related diagnostics that the company offers. These are the objectives for the presentation. We want to learn a little bit about the interplay between both the innate system and the specific immune system. Those are important distinctions. The innate immune training versus specific immune memory, especially as it relates to things like COVID and micronutrient deficiencies and overall health function, and celiac and the difference between celiac and non-celiac gluten sensitivity. Then I'd like to spend some time explaining a bit about a newly discovered role of how innate immune cells create a defense against pathogens, but how, like any innate response, can be a double-edged sword. Neutrophil extension traps, or nets as they're called, can both be effective in defending the organism, but can also be deleterious and can cause autoimmunity, aging, and now we learn fatality in COVID, related to COVID. There's speculation that nets are related to metastases of cancer. We want to discuss food and immune activation, the advantages of a cellular test versus a serum test, micronutrients and immune function, and especially in the context of a current pandemic, and the advantages of a cellular approach to testing micronutrients versus a serum approach. And of course, overarching this whole issue has to do with immune function. This is the cover of Science, as you can see, several years ago, 2013, January 11th. And what we're looking at is a lymphocyte and a specific immune cell. And what they're showing superimposed on that, I'm sure you'll recognize, is a yin-yang sign. And the yang is the inflammation component, or the attack component, or the defense component. But then equally important is the yin compartment, or the yin functions of the immune system. And they're equally, I would say, present within the body. Hopefully they're even more present. That creates immune homeostasis. Today, however, both need to be in balance. It's not a question of just pumping up the immune system, because as we see with COVID fatalities, it's the people who have the most overactive immune system that suffer the strongest consequences. Then it's the people with depleted immune systems who are next in line, who may not die, but have the more severe cases. But what we want to achieve is balance, and that's been an overarching theme in our laboratory development over the last 35 years, our laboratory testing development. With respect to COVID, there's information now that COVID fatalities are associated with neutrophil extension traps. And what's been found on autopsies in the microvasculature are immunothrombi, which are comprised of these nuclear neutrophil extension traps, along with activated platelets. And others have reported the same thing, that in about 10 or 15% of patients that progress to serious pneumonia, there's a little known yet powerful function of neutrophils. Of course, neutrophils are the main cells of the innate immune system. And the ability to form neutrophil extension traps may contribute to organ damage and mortality. There's a number of other references, and of course this will be available to everybody, so one can see the targeting potential drivers of COVID, neutrophil extension traps in COVID-19, on and on, oxidative stress neutrophil extension traps. So it's kind of a big deal, and I think we need to probably pay more attention to it. Does diet have anything to do with it? Does diet have anything to do with COVID and everything else? Well, I think it's kind of a rhetorical question, but it's kind of interesting to see research coming out in these publications in paper journals like Cell, where they talk about how today about 80% of non-communicable diseases are associated with aging, are caused by a Western type calorically rich diet. These disorders, of course, are diabetes, mellitus, obesity, cardiovascular disease, things that we're quite concerned with. It is well appreciated that disease progression is strongly associated with inflammation involving the cells of the innate immune system. And it is conceivable that these sterile inflammatory triggers, such as found in the Western diet, can induce trained immunity. So trained immunity is different from memory that we hear about. Like a vaccine exposes somebody to a pathogen so that we then remember it and produce antibodies against it, or mount a specific immune cell attack against it on reoccurrence. We want to develop memory. Training of the innate immune system is different. The difference lies in the fact that training induces changes in the epigenome so that the cells will become more responsive to other stimuli. In fact, they state in their paper in such a scenario, a secondary stimulus would then trigger altered and potentially pathological immune responses. So I do think that there's good reason to believe that a less inflammatory inducing diet that doesn't cause the innate immune cells to get into an aggressive posture, which persists for quite a long time. Again, change the epigenome, which changes the expression of the progenitor cells of the innate immune cells. And those cells are primed, and then another trigger can set them off. So what are these NETs? Neutrophil extension traps. It's a new mechanism of innate immune-mediated tissue injury. Of course, you know, innate immune cells destroy pathogens through phagocytosis, through generation of free radicals, through dosing them with toxic mediators like TNF-alpha, IL-6, so on and so forth. But then there's a new mechanism that was discovered in 2004. And if you Google NETs and NETosis, you'll see that in 2004, 2005, there was hardly any attention to it until it was discovered over at in Berlin at Max Planck. And then each year, the number of papers has increased exponentially. And it's kind of interesting that also NETs pertains to neutrophils. Again, the most abundant innate immune cell in the periphery. It's interesting also, these are short-lived cells, and about 100 million of these cells die every day naturally in a resting state through apoptosis. That represents 132 million miles of DNA. Now that DNA, if it's coming from cells that are in a resting state, is actually anti-inflammatory. In fact, transfusion of apoptotic lymphocytes and neutrophils is used to prevent graft versus host disease. So that's all fine, but what happens if again, Western diet, trained immunity, other triggers are there, and we have necrosis or pyroptosis, these other kind of responses? Then we have more DNA that is in a different condition. We'll kind of circle back on that a little bit. And that can drive autoimmune disorders, particularly arthritis and lupus, preeclampsia and metabolic syndrome, and other autoinflammatory conditions as well as cancer, and as well as we've seen a major driver in COVID fatality. Remember, the total amount of DNA in the system is about twice the diameter of the solar system, which is kind of remarkable. So it's overall cell-free DNA. You'll read a lot about it. You'll hear a lot about it in the present and the near future as a way to make diagnoses of cancer and so forth. That along with methylation patterns. This is what it looks like. So this is actually a neutrophil which has undergone netosis. And in this process, what we're seeing as a benefit is that the nets ensnare bacteria, and then contents that were within the cell that are now within the nets destroy the bacteria. So this is a valid defense maneuver that's the good edge of the sword of the innate immune system. Now, when the process is over, something has to clean up that mess. And remember, again, we're talking about a lot of cells and 100 million a day normally, but they're not undergoing netosis all the time. But again, what I want to emphasize, or what I want to connect here, is the idea that food sensitivities or other triggers, other inflammatory triggers, can induce the process of netosis inappropriately. Now, normally this degree is mopped up by enzymes, DNases. However, you can imagine if we have a large number of cells undergoing processes like this, and when they occur and the DNA is freed from the nucleosome and becomes part of the net and then is out in the circulation and it's not cleaned up, this drives autoinflammation, this drives autoimmunity, this may drive cancer, and it drives fatalities, immunothrombi, and critical tissues that result in COVID fatalities. So where does it come from? What are these nets made out of? This slide I'm using with the permission of my friend Joe Belanti. Some of you may know, he's presented at your fellowship. Inside the nucleus, the DNA, remember there's a lot of DNA, 6.7 billion miles of DNA is tightly wound up around these octamer proteins called histones. And the reason is that this keeps the DNA suppressed from expression, generally speaking. One of the components there, CH3, can then undergo an epigenetic modification through the acetylation of an extension from this histone. There are eight such molecules in each histone that has a lysine tail. And when it's acylated, that changes the charge of the histone from being positive to being negative. Of course, the DNA has a negative charge. And this loosens up the DNA so that it can become exposed to proteins that will cause promotion of gene expression. So that's how some epigenetic modifications work. It's quite normal. It happens billions of times, hundreds of billions of times, probably every second, that we have some kind of change in genetic expression. But these histones themselves become part of the constituents of nets. As the breakdown happens, there's a decondensation of the nucleosome, and along with the DNA that is expressed in the nets, there's also histone molecules. These are extremely toxic. These are factors that are intended to kill. They have a dual role, obviously. We talked about their normal function, but they also function to kill pathogens. Just think what they do to our own cells when we have that going on. So what does that have to do with the price of beans? Or what does it have to do with living? we had collaborated a few years ago for a period of years with researchers at Yale, and this group, Ali and Garcia Martinez, undertook a few clinical studies and also some technical studies. And one of the things they found out, and they reported in Nutritional Metabolism in 2018, was that foods can induce the release of DNA from cells. And they did this by running Alcat tests on patients, finding out which foods would produce a positive Alcat test or induce a neutrophil response and other granulocyte responses, and then look into the supernatant and do an analysis for the presence of DNA and compare that against background counts and foods that did not produce a positive Alcat. And what they found was that there were greater quantities of cell-free DNA following a positive Alcat test. Now, I just want to mention a little nuance here. They did not determine whether the DNA came from the mitochondria or the nuclei, because the mitochondria has 47 genes, and they kind of look like, and mitochondria kind of act like prokaryotic cells. They don't have a nucleus, and there's much fewer methylation sites than there are in the nuclear DNA, where most of the genes are switched off because the cell's fully differentiated. They only need a few genes. And the reason that's significant is that in general, when you're talking about naked DNA, that coming from the mitochondria is much more inflammatory than that coming from the nuclear sources. And so that gave rise to some research that we've undertaken, and now concluded that was funded by GlaxoSmithKline, to determine whether the release of DNA seen in a positive Alcat response is from the mitochondria or from the nucleus. And we'll be reporting that hopefully in the next couple of months. So the conclusion is that this is the first demonstration that foods can result in the release of DNA that we identified by our test. This may provide, and this is an interesting, I think, very important conclusion, this may provide a mechanistic rationale for the reported findings and clinical improvements in patients using leukocyte activation tests. And here, that's the generic name for the Alcat test, to guide dietary choices and provide support for conducting randomized clinical trials for these tests in GI and liver diseases. And that in terms it does. The operative thing here just provides a mechanistic rationale. For years, we've been seeing patients with complicated issues and some autoimmune issues which keep going up. And we identify a few trigger foods, they avoid them, and the symptoms go away. And so, we know some of this is driven by free radical damage, and some is driven by toxic mediators, and they're all sort of connected. It's a very complex, orchestrated response. But now we know that fundamentally a great many of these more complicated and difficult to identify a source conditions that people have, which so many of your patients have, could be this cell-free DNA. And these are the kind of conditions and these are the kind of responses that are affected: metabolic, GI, skin, migraine, respiratory, and our joint, and basically any part of the body can be involved because the neutrophils are all over the place. And essentially, the schematic here shows a resting cell being triggered by a receptor, which could, this could be on the surface, it could be a toll-like receptor. And it causes changes in the cells flatten out, the cells become active, the mitochondria turn on, produce free radicals that interfere, that triggers net formation, and we have release of reactive oxygen species, nets, and all the different components there, including that we've talked about, resulting in these kind of outcomes. It can affect the heart, GI, respiratory, so on and so forth. Looking, again, interestingly, trying to be a bit translational and look at how other disciplines support this endocrinologically. Guys from Cold Spring Harbor, actually from Stanford, Odegaard and Shala, reported in Cold Spring Harbor Perspectives in Medicine, looking at the cases of type 2 diabetes, that it seems to be an innate immune response that drives that the surplus, and by this they mean the increasing, and at the time that paper came out, they said that the diabetes had doubled in 10 years. Now, this was the doubling occurred in the autoimmune form of diabetes, type 1. I'm sorry, in the type 2. In the type 1 condition, which is the autoimmune version, the HLA version has been more or less consistent over 20 years. Now, what is HLA? And what is type 1? That's not that's juvenile rather than so-called adult. Well, now those two categories are kind of melding together into diabesity. But HLA stands for human lymphocyte antigen, which means that the specific immune cells recognize their own tissue related to islet cells and inflict immune-mediated injury and destroy the tissue, and therefore the gland cannot produce sufficient forms of insulin, as opposed to the acquired immunity of too much stress, too much sugar, and downregulation of receptors and inducing insulin resistance. So that's an interesting thing, that overall inherited causes of diabetes have been relatively active. And I just want to give some context here. When we talk about HLA, we're talking about this molecule here. And this is the process that is common to specific immune system activation, which is distinct from innate immune system activation. First, an antigen is taken up by an antigen-presenting cell, which digests the proteins, breaks it down, and those peptides merge with a molecule that transports it to the cell surface, where it then is recognized only by certain T cells that have this kind of T cell receptor that recognizes that particular antigen complex. Those cells then divide and then either become cytotoxic lymphocytes that destroy tissue directly, or send signals to B cells that recognize that those molecules, and then they divide and produce antibodies that go into the circulation that fight the inciting pathogen and enable, it's not depicted here, enable phagocytes to then attach to this pathogen and consume it. That could be an allergen, it could even be self-tissue. So the whole thing is dependent upon this interaction here. And if that genetic predisposition, which is a human lymphocyte antigen, does not exist, or does not have, it doesn't exist, then it doesn't produce that molecule, and none of this stuff happens. And of course, the whole concept behind memory is depicted here. Once it happens, when the inciting stimulus is perceived again, it causes this process to occur much more frequently. The cells divide much more quickly, the antibodies are produced much more rapidly, and the problem is hopefully resolved much more quickly. So what's interesting is that the increase in type 1 diabetes by genetic risk is been in the medium risk group, in other words, people who have those genes, has doubled. In the low risk group, it's going up 3 times. In the people who don't have that risk at all, it's going up 7 times. The conclusion I get from that is that there's another mechanism going on, and that is innate immune system is driving damage. And we see from other research, also from a reputable journal, Cell, that it's extracellular trap formation, particularly unmethylated CpG islands, cytosine phosphate guanine, which are unmethylated. Again, those are the more pro-inflammatory DNA or RNA acids, drive insulin resistance. In mice that are fed a high-fat diet, there's an increase of extracellular traps in the vascular adipose tissue and decreased clearing, and then increased autoantibodies against conserved nuclear antigens. There you go. Here you have a pathway that explains why even people without the genetic risk can develop what previously were thought of as only inherited diseases like diabetes 1. So again, another perspective on the significance of diet and maintaining the yin and yang of the immune system, not having these chronic insults in the form of toxic foods. So this relates to the Alcat test. I want to try and cover, if possible, all these tests in the time we have left. The Alcat test is a challenge of the innate immune system. So the purpose is to find out what are the triggers of food sensitivities. Not allergy, which would be specific immune system, but delayed type non-IgE mediated realities that cause chronic symptoms, impairment of cell function, and so forth. It's a whole blood test. It is not a serum test. We are not looking for antibodies. We are not looking for particular proteins. We are measuring the whole cell in the context of the whole blood, where all the different subsets of the leukocytes are present, even the red blood cells, although they're not much of a factor. We lyse them before we make our measurements. We do an ex vivo challenge while the cells are alive, and we measure the effect of the stimulus, the food or the herb or the chemical or the functional food or the pharmaceutical substance against the blood. And how we do that is that we use a machine. You can see here, we get these graphs, and I'm going to explain those in a second. We get these graphs, and this graph correlates with these microscopic images. A negative graph shows that their cells are not reactive. A strongly positive graph where there's a loss of cells. This is what it looks like under the scope. And if there's a change in the cells that doesn't cause complete degranulation, it looks a little bit differently. How do we get those things? How do we get those graphs? So we have a device which takes the whole blood, challenges it with an allergen or not, where we use those samples as a control, and we pass it through. We have a hydraulic system that passes it through an electromagnetic field, which is sensitive to the change that the presence of the cell induces in the electrical field. And so we're able to generate a size distribution curve of the leukocytes. And this would be the generation, that red line would be the graph when there is no substance added. That's the baseline. And the overlaid blue would be a positive response, showing what happens to the cells when there's a reaction, and the cells are degranulated and they have released their contents and may be primed for that pyrotoxics, or not many, in many cases they are, will progress to the formation of nets. So we don't presume a particular mechanism. We don't presume that there's an antibody involved. Maybe there is, but I doubt it. We do know that complement proteins contribute to this. We do know that the cells contain a lot of receptors, internal receptors, non-like receptors that are sensitive to DNA fragments. There are toll-like receptors that respond to DNA fragments. So there are many different pathways. There are receptors that respond to pharmacological stimuli, meaning the toxins that naturally occur in foods. All plants contain natural toxins to protect themselves. So whatever it is, we don't know. We really don't care. It's not specific. We do know that whatever it is, it's creating a cellular response that's producing changes that we have now identified as being associated with NETs, and also release of other toxic mediators. And by the way, so many people still rely upon IgG, but I think you should, if you do, you should consider switching to Alcat, if I may be so bold, because it's been in the literature, but unless you're an immunologist and you search literature all the time, you may not know it. But IgG antibodies are normal, and they're there to help the body eliminate excess antigen in circulation by the monocyte macrophage system without inducing inflammatory responses. And we know, what do we do for looking at COVID exposure? IgG antibodies. So if you have IgG antibodies, you may have it in your system, but it may be gone from your system. You still have IgG antibodies. That's what gives you the immunity. So it shows you exposure and it shows you tolerance, not sensitivity. So that's the main point I wanted to drive home with the Alcat test. The test results are pretty comprehensive. We test for as many as 470 substances comprised of foods, food additives, environmental chemicals, antibiotics, anti-inflammatory drugs, molds, medicinal herbs, functional foods, and supplements. The reports are pretty comprehensive. If there's reactions to gluten, for example, reflex, other grains that contain gluten, it gives a 3 to 6 month rotational elimination guidance. And as a matter of fact, you can also, anybody who takes a test, a practitioner or your patient, can get included in the test price a free 20, 30-minute private one-on-one consultation with a dietitian or dietetic counselor to go over the results just on the Alcat test, and that would be it. If you want that, it's there free of charge as an option. There's a study which is ahead of publications, going to be published next month, showing the different changes in BMI, scale weight, and waist measurement amongst 4 groups. Alcat, this is from University of Texas, Galveston, Buck Willis and his team. Food allergy elimination plus aerobic surge exercise, Alcat avoidance alone, exercise alone, and neither of them. You can see that the best is to exercise along with having a healthy diet. But just having a healthy diet is the biggest principle. Another recent study published in BMJ from some work at Yale was with patients with IBS, and we looked at global improvements in scale and symptom survey. And this is the treatment group. It was a prospective, randomized, double-blinded, placebo-controlled. The placebo was a sham diet, which was based on Alca Test, but left in positive foods. But when there was a positive food in a certain category, it would eliminate another food from the same category to maintain the same rigor and complexity of the experimental diet. So it was very well controlled, and you could see a sharp drop in symptoms after 4 weeks, even greater after 8 weeks. Amongst both groups, but the delta was phenomenal. So there is placebo happening, and I think if they left in the category-related foods, the differences would be even better, because due to cross-reactivity, that feature probably, although it was good for experimental design, probably mitigated against the outcome. But the outcome is still remarkable. Again, we've looked at that. So that's the Alcat test. We have a few more minutes. I want to speak about our cellular nutrition assay, which is also a functional test rather than a serum test where we're looking at static levels in the serum of particular micronutrients. Here we're looking at how the cells respond to the addition of a micronutrient in a cell culture versus a baseline, which is identically treated, except the one single isolated variable is the absence of that test substance. So again, this graph, lymphocytes, specific immune cells, quell an infection, they need to divide. In order to divide, they need nutrients. So what do we look at? We look at lymphocyte counts. Lymphocyte counts in general, under non-experimental conditions, have been shown to be very predictive of mortality. A low lymphocyte count is not a good thing, especially in relation to a high neutrophil count, and you can probably understand why. This is a recent article just a year ago in JAMA. When you have inflammatory markers to boot, it's even worse. And I won't go into the details of that just in the interest of time, but there's a reference. Where do we get the idea to look at lymphocyte proliferation as a marker for micronutrient sufficiency? It came back from the 40s. An experiment was done by these folks at George Beadle and Ed Tatum, University of Chicago, looking at a basic common mold spore that grows in bread. It's orange. They cultured it in central growth media. It replicated fine. They calibrated the media to just make it sustain itself. Then they irradiated the samples of the media, and they found that they arrested cell growth. And they realized that they disrupted through the X-rays some metabolic pathway, which was essential for cell growth, and they added back nutrients one at a time to support that, and eventually found that by adding back arginine, they were able to restore cell division. So it's kind of like when you have another inborn error of metabolism as opposed to one induced by X-rays, like methylene tetrahydrofolate reductase alleles that we hear about a lot. You can support those pathways with B vitamins, B12, 6, and 9. So the cellular nutritional assay has 3 components. One is to look at lymphocyte proliferation when we add a micronutrient. Another is to look at the status of the ability of the cells to resist oxidative stress, use hydrogen peroxide as a stressor. And then we carry out that same assay, but then we add in certain antioxidants to see how well they protect the cells against the oxidative stress. And of course, just as a reminder, metabolism happens inside the cells. This is a list of the currently the items that we test for in the micronutrient assay. The procedure, we separate the patient's lymphocytes, add back autologous serum. Now that's kind of different than what SpectraCell developed, and I was very familiar with their work because I worked with the inventor of that test many years ago, William Shive. And without going into a great deal of detail, although we have a webinar about that we can send to anyone, it's a major difference because Shive's attempt was to standardize the cell culture medium without fetal calf serum, which would be a variable from lot to lot. And so he developed an artificial cell culture medium, which was brilliant, and it did support cell growth, sort of, to a degree, but not perfectly, but it was standardized. But we innovated, and I know why he wanted to do that and didn't want to take my suggestion to use the patient's own serum. It was a different factor driving that decision. But we found that there's nothing better than the patient's own serum. That's the most in vivo-like thing and provides the growth factors. So we take the lymphocytes, separate them from the other blood components, add back the serum, stimulate the cells with the mitogen, mitosis generator. We use PHA. And then we get a baseline measurement of proliferation rates. And we use optical techniques to look at metabolic activity, which correlates with proliferation rates. So we don't use any radioactivity. And then we culture in the test wells from the same sample, identically treated, subsamples that are exposed to a micronutrient. And then we look at how that micronutrient has improved the proliferation of the cells. And if we find marked improvement, then the logical inference is that regardless of what there is in the serum, functionally, for whatever reason, it's not sufficient to produce optimum proliferation because we're inducing proliferation with the mitogen, so we want to see it. And that's how the report comes out. If there's not much induced proliferation, it's green, and then if it's in a borderline level, it's yellow, and then we give a number and we record it, in a red area. So those are things to consider for repletion, preferably with foods, but can be also supplements. These factors impact, of course, cellular metabolism. The redox assay is reported as the person's ability to resist oxidative stress in relationship to normal ranges that we established by testing a number of subjects. These are the items that are in our antioxidant protection assay. And it's reported similarly. If we find that the addition of a particular, let's say, pycnogenol or noni berry or milk thistle protects the cells from oxidative stress, consider using that. Again, the same kind of report structure. We also offer testing for methylation, which is very important. There's a lot, this is the methylation pathway. These are the genes that we measure, and we look for alleles, variants that are associated with inefficiency. And in our report, it's a smart report that can give, if a person is heterozygous or homozygous, it gives either a yellow reaction or a red reaction, or if they're homozygous negative, a green reaction, where that is in the pathway and what you can do about it with the various the B vitamins. Of course, methylation is critical for a lot of things. Also, poor methylation results in overabundance of homocysteine, especially if people eat a lot of dairy and meat. There will be a lot of methionine, and then if there's deficiencies in the B vitamins, and there's genetic variants, particularly in MTHFR and the others in the pathway, there could be an abundance of blood levels of homocysteine, and that's toxic. And that's been shown to be associated with damage to arteries as well as other tissues. So this is a list of symptoms that are associated with that. The results we've gone over. Homocysteine on its own, we look at that. It's good to do that in conjunction with the test, with the methylation. Almost done here with the testing, and we'll move to questions. Zika, celiac IBS, and Crohn's array. You're not what you eat, or you're what you assimilate. Of course, inflammation in the gut leads to leaky gut, the foundation for food sensitivities, malabsorption. It could be local inflammation, so forth. Not all celiac is, non-celiac gluten sensitivity is, non-celiac gluten sensitivity, forgive me, it's not all celiac. There is something called non-celiac gluten sensitivity. Again, think back to the early part of this discussion. If you have the genes that produce the MHC molecules and the T cell receptors, particularly the MHC molecule, that recognizes gluten peptides, it can activate T cells when activated and then exposed to tissue transglutaminase, an enzyme from damaged enterocytes, combined with the gluten peptides and the right genetics, the T cells go bonkers and eat up the villi, and you have celiac disease. If you have the same phenomenon, but you don't have the genes, you have non-celiac gluten sensitivity. That's why the genes are something we test for. Non-celiac gluten sensitivity has been proposed to exhibit an innate immune response activated by gluten. Of course, in the celiac, it would also be innate and specific. Some researchers at the University of Camerino in 2018 reported that because, this is a big thing in Italy, especially with Fasano's group and everybody kind of thinking that they have to avoid celiac, and Fasano's group getting a tad, and people he worked with in Italy getting a tad defensive about the fact that everybody's talking about leaky gut and non-celiac gluten sensitivity. They brought out the idea that the way to determine that is not just relying on your own subjective evaluation by avoiding gluten and seeing if you feel better, which was the diagnostic test. They said you have to do a double-blind oral challenge. So one of the groups we work with at University of Camerino looked at testing 5 grains through Alcat and double-blind oral challenges. These are the results. They were identical to a double-blind oral challenge. And if you it was 64% concordance, but they only looked at 2 expressions, abdominal pain and bloating. I think a lot of these things are silent, a lot of things produce distal effects. I think had they included those kind of criteria, we would have seen a higher degree. These are kind of common symptoms: GERD, canker, everything. This is what we look for. We look for the HLA, human lymphocyte antigens, DQ2.5 and DQ8. Now again, fantastic negative predictability. If you don't have these, you don't get celiac. If you do have these, do you get celiac? Maybe. Other cofactors have to be there. We look at markers for genetic markers for Crohn's. We look at serum markers that are associated with tissue damage. We look at total serum IgA, because if there's an IgA deficiency, that's not good, but it also means you have to alter your perspective on looking at the deaminated gliadin peptide and TTG, which are markers of direct tissue damage. Antibodies against Saccharomyces cerevisiae are predictive or associated with Crohn's. And of course, we look at, optionally, at the foods, at the sensitivities. So this would be kind of an algorithm for determining how to interpret that. I'm going to leave off here. It's very interesting why we test these particular genes. They have to do with sensing of pathogens and autophagy, as well as antigen recognition. And the clinical significance of those have been established. The last test that we offer is telomere length. And of course, inflammation drives shortening in the telomeres. And when the telomeres gets short enough, the cells become senescent. They don't necessarily die, which is a bad thing because they're causing problems. So that is a marker of what a person's biological age is. And we report that again when we do the redox, where the person sits in terms of their age range according to normal standards that we've developed. This is our motto, live food without medicine. I think someone said that before. And I wish to thank everybody for patiently participating in this. And I see so many of you stayed. So this is our information if you want a copy of this. I'm not sure if you contact us or the A4M, but if you have any questions, here's our contact information. In the last few minutes, I'm here for any questions.
Excellent. Thank you so very much, Roger. As a reminder to all of our attendees, please submit any questions in the Q&A box within your Zoom window. Getting started here, our first question, is it DM type 1 or type 2?
Diabetes mellitus type 2? Is it, well, what I was talking about is you know, they kind of have seen that they blended together. And what I was pointing out with the work from the people at Stanford was that the type 1 is increasing amongst people who do not have a genetic risk tremendously. So why is that? So it's not just the understood pathway of T cells destroying islet cells because there's an antigen recognition phenomenon. What I showed in that next study after that was that innate immune cells can start destroying the pancreatic tissue and produce what looks like or functionally is type 1 diabetes. Never mind that food intolerance causes all kinds of issues that create the grounds for type 2, which is insulin resistance. So it's really both. And I know it's a bit subtle and nuanced, but if you think about it, I think it'll make sense. I hope I answered that question.
Is the CMA test time sensitive for travel refrigeration?
No, we need whole blood. We're good for 72 hours, but we provide, as we do with all tests, mailers that have requisition form and of course the protocols in them, the collection information, and we use 2 sodium citrated tubes that do not need to be refrigerated, but it's better if it gets to us overnight. And all the mailers contain a prepaid FedEx air bill to do that with.
Are there further validation studies on the Alcat and any for the nutrient test? Curious to know sensitivity, specificity given IgA, IgG testing is notoriously poor in this regard.
Yeah, I completely agree. Our IgA and IgG testing for food sensitivity is pretty much useless, and I've given talks about that because it's normal. We have a scientific dossier, which is on our website. You can download it, and it contains about 50 research papers and published studies on validation of the Alcat test. And there's one study published on our CNA also on our website in Journal of Functional Foods. And in that study, which unfortunately we didn't get to talk about today because of time, it shows that there are a greater number of micronutrient deficiencies when people have a greater number of food sensitivities, and there are a greater number of micronutrient deficiencies and lower redox potential amongst people who have poor lifestyle habits like smoking and so forth. So a nutritional test is a little bit more difficult to validate, but so far that research suggests that we're on the right track with what we're doing.
All right, we do have a hello for you, Roger, from Killalow, Claire. Roger, fantastic to see the advances in the Alcat test over the last 20 years.
I'm sorry, I didn't get that.
It was just a comment. Fantastic to see the advances. Alcat test over the past 20 years. Pascal and Frida Carmody.
Oh, Frida. My God, it's been a long time. It's so delightful to hear from you. I hope you're doing well. Thank you very much for that.
All right, any last questions we'll take now before we roll into our next lecture? Thank you so very much again, Roger, for an excellent presentation. We appreciate you being here with us today.
Great. It's been my pleasure. Thanks for everybody's patience in listening.
And thank you to Cell Science Systems. Thank you.
You bet. Thank you very much, Rudy.